So it's also my pleasure to introduce our last speaker for today, who's Dr. Richard Glynn from the Genomics Institute of the Novartis Research Foundation. Richard went to Cambridge University and received his PhD at what's today the Cancer Research UK under the direction of John Truesdale in immunology. He did a postdoc at Stanford with Chris Goodnow, working B-cell tolerance, but also really pioneering microarray techniques in this field. He went on to work in biotechnology, in genetics and genomics, here in the Bay Area at first at a company called EOS, which was in South San Francisco and then ultimately acquired by Protein Design Labs. And then moved down south for a couple of years at a company called Phenomics before joining GNF. And at GNF, that's when he was coaxed into infectious disease by somebody who many of you might recall was another Berkeley faculty, Pete Schultz, who's the illustrious director of GNF. And they had the bright idea to bring modern drug discovery methods to some of the most important neglected parasitic diseases that Richard will talk about today, and doing so under the banner of a multinational pharmaceutical company. So really pleased to have him come and talk about his work. Thanks, Richard.
All right, thank you for inviting me today. I'm delighted to be here. I came here to talk about Kinetoplastid diseases, and I was expecting to give you an introduction into these diseases, which don't get a lot of airtime and are not particularly well known and high up in people's consciousness. I'm not sure I got that right. Anyway, but then I arrived here and I saw that in the T-shirt collection outside, actually at Berkeley, you have trypanosomes on T-shirts, so which I've never seen before. Made me feel very at home. Thanks for that. But this collection of diseases, there's three main ones that that we've been focusing on: sleeping sickness, which we're collaborating with our colleagues in Singapore in the Novartis Institute there, and then Chagas disease and Leishmaniasis, which are the two diseases that I'll be talking about today. So this title slide has a triatamine bug. These are in real life about an inch big, so that they're pretty big, not this big, but they're pretty big, and they carry the causative agent for Chagas disease, which I'll go into in some detail. But they're very Chagas disease is very prevalent in Latin America, but it's also important to note that because of emigration out of Latin America, there's quite a prevalence in the US and in Spain, and there's also actually endemic transmission, not to people, but to primates, at least, in Southwest, in Texas. So that you can catch Chagas disease actually in the US. So let me tell you a little bit about these diseases and what they have in common and why we thought it would make sense to kind of tie them into some coordinated drug discovery program. The main thing that really struck our attention was that there's been very little drug discovery or drug development in this area for many years. Most of the drugs that we use to treat all three of these diseases were developed 50 years ago or so. And the other thing that's striking when you look at the list of drugs that are used to treat these diseases, very few, possibly none of them, were actually developed to treat any of the parasitic diseases that they're now used for. Instead, they were repositioned from other diseases. And when that happens, it's great because it's a very fast path through development. But on the other hand, you end up with drugs that are not particularly perfect and don't meet the target product profile. They're sort of good enough to be useful, but maybe not better than that. So this is what the parasites look like. You can see on the left-hand side, these are trypanosomiasis. They look familiar probably from the T-shirts. This is the pathogenic agent for sleeping sickness, T. brucei, and then on the right-hand side, T. cruzi. You can see that they're flagellated. They get their name as a family from this Kinetoplastid organ, which is full of DNA. It's like a mitochondrial-like organelle and stains here at the base of the flagellum. And I don't know if this will work, but I'll give it a go. And this is not, well, okay, imagine them wiggling. Then And then the other thing that kind of gave us encouragement that it would be worth diving in all three diseases in parallel was that the genome sequences are sequenced, and we know there's lots of homology between the three genomes. Roughly half the genes you can see paralogs from one species to another. And that made us think, well, maybe we'll be able to find compounds that, in the best of cases, we can bring forward for two diseases or maybe all three diseases in parallel, each one de-risking the next one, of course. So I'm going to spend the next part of the talk focused on Chagas disease. As I mentioned, Chagas disease is an endemic in Latin America. It's not very well known, but there's estimates of 8 to 10 million people that are infected with the parasite. And the etiology of this disease is as follows. Usually kids get bitten by the triatamine bug that's infected with the parasites. There may be some swelling at the bite site. You can see this picture here of a girl who was bitten near the eye. These bugs are also called kissing bugs. And they're called kissing bugs because they tend to kiss or bite on the face whilst kids are sleeping. And then there may be some fever and a bit of swelling. And then after a couple of weeks, the immune system kicks in, controls the parasitemia. And then essentially nothing happens. And nothing happens for decades. And in many cases, people will die of something else before any other complications from the disease manifest themselves. But about a third of the people suffer from Chagasic cardiomyopathy or gastrointestinal complications. And you can see the parasites here forming a little nest in the heart. And it's that population of the 10 million already infected people who are at a 30% risk of developing this disease, which can be then fatal, that we were trying to figure out, well, can we find a drug that will mop up the last remaining parasites in these chronically infected patients? The parasite burden is very low, but still sufficient to cause disease. The incidence of Chagas disease through the world is shown in this map here. You can see, mainly driven by where the parasites are endemic, which is Latin and Central America, and then emigration of Spanish-speaking people to the US and to Spain and Australia is spreading this disease through the world. So as far as how it's treated at the moment, there's been two drugs. They were developed in the 60s or 70s, benzonidazole, nifurtimox. Benzonidazole's used the most often. It works reasonably well in acute infections with kids. It's reasonably well tolerated. As people get older, the tolerability of benzonidazole goes down or seems to go down. And in particular, it's associated with a very dramatic dermatitis that can lead to treatment discontinuation. In fact, only recently have we had double-blinded, placebo-controlled trials testing the efficacy of benzonidazole. And it was actually very interesting if you look at the ability of benzonidazole to clear parasites in a persistent way. When those patients are treated per protocol, which is a 60-day treatment protocol, is actually quite effective. 90% plus of patients were parasite-free 6 months after treatment. However, if you look at the intent to treat success rate, it was only about 60%. And that's because a lot of patients don't finish the course. So that was where things stood in 1970 or so. It's pretty much where things stand at the moment. And when we got into this game, we thought, well, what can we do to try to contribute to improving this situation? And when we looked at the landscape at the time, which was about 5 or 6 years ago, there was a lot of excitement around CYP51 inhibitors. CYP51 inhibitors that have been repositioned from the antifungal field, particularly Posaconazole, which was a Merck sharing drug, and E1224, which is a prodrug of ravuconazole that DNDi were developing with ASI. And that was really the only target for which we had good in vivo evidence and potent compounds, preclinical in vivo evidence and potent compounds that looked encouraging. Now, we were, again, struck by this kind of repositioning approach to treating tropical diseases, which is exactly what was repeating itself out with the antifungal azoles. And so we thought, well, what if we take an unbiased approach, run a phenotype-based screen, try to measure the growth of these parasites in a 1536 well, 10 microliter reaction, and see what the data tell us. Is that the best target to go after, or are there other things to go after? So we developed some assays, firstly using a beta-gal tagged parasite that we got from Fred Buckner at UW, and we were very grateful for that. And then on our secondary assays, using high-content imaging to measure the proliferation of parasites inside host cells. These are 3T3 cells, and you can see the tiny green dots are the nuclei of the parasites proliferating within those cells. So we ran this screen. We looked at our most potent compounds, and sure enough, what we found after going through all this effort was that the most potent compounds actually hit CYP51. So I thought, okay, well, we better follow the science. Is there any reason for us to believe that we can make a better CYP51 drug against Chagas disease if we make one that's kind of built to purpose rather than repositioned from another field? And so we started down this path thinking maybe we can fix some of the drug interaction issues of Posaconazole, maybe we can find a more potent compound, something that we could formulate as a tablet, and so on. And we started here. This was our starting molecule, incredibly potent in a cellular assay, 5 nanomolar off the deck. Usually drug discovery programs don't start off this well, so we were pretty psyched. There was a structure available. Our chemists worked together with a structural biologist to build our hit into this groove here. And as we built into the groove, you can see a nice drop in potency now, sub-nanomolar, 200 picomolar cellular potency. Things got even better. Now we're at sort of 50 picomolar cellular potency. So we were very, very excited. We had less CYP interaction with human CYPs. We thought we may have a better profile than Posaconazole. And so we thought, well, we optimized the compounds to go into animal models. And this is what happened. So things still look good. Here's the compound here in the groove, 50 picomolar cellular activity. We set up an acute infection model where we infect mice with the T. cruzi parasites, wait a day or so, treat with compound, come back after another 5 or 6 days, measure the parasite burden in the heart. And you can see quite a nice 3-log drop, both with our lead inhibitor and with benznidazole and with posaconazole, which was, as I told you, moving into clinical trials. So that seemed great, and then we knew that the patient population that we were really focused on are not people with acute infections, but people with chronic infections. And so we built a model to model chronic infections of Chagas disease. It looks a little bit like this. So you can see the parasite load here. We infect here on day 0. You can see this little burst of parasitemia, and then after a couple of weeks, as I told you, the immune system kicks in and works in the mouse as well as in the human. Parasites come down more or less to baseline. And we wait for that to happen, and only then do we treat. And if you treat with a drug that works, benznidazole works in this model very nicely, then you see that even after you immunosuppress the mice to allow any parasites that are remaining in the host to proliferate and come out into the blood, even if you treat with benznidazole, you just don't see that, right? So these mice are what we loosely call sterilized or cured of the infection, at least judged by blood parasitemia after immunosuppression. If you don't treat them, you get this massive burst of parasitemia that you can't leave for too long before the mice get very sick. So we had a model that, and I should say, we didn't invent this. It was really driven by information in the literature, but we had a model in our hands that we thought would allow us to test our compound in a chronic setting. And let me just walk you through what's going on here. So here you can see the groups. We have vehicle-treated mice, and then benznidazole-treated mice at 3 doses, and then our lead inhibitor at 3 doses. So when you look at the end of treatment, you can see that with benznidazole at increasing doses, you can drive the parasite load down below the limit of detection by PCR. And with our lead compound, you can do the same thing, which was great. The problem came when we immunosuppressed the animals. And when we immunosuppress the animals, what you can see is that the benznidazole-treated animals at the highest dose remain parasite-free. That's good. That's how it should work. But the CYP51 inhibitor that we developed, that we were very proud of, with the 5 picomolar, 50 picomolar activity, basically no activity after immunosuppression. All the animals rebounded, every one of them. This was really not a good day, but I think it's good to share failure as well. So we thought, well, is it our compound or is it the target? If it's our compound, maybe we can make a better compound. If it's a target, then it should be a shared phenotype that we would see with Posaconazole too. And so we repeated the experiment, and to keep you and I on my toes, the format of this graph is slightly different. Here are the groups, and then the post-treatment animals are shown here, parasite load, and the post-immunosuppression are shown here. And so what you can see here is that if we treat with benzonidazole for 10 days, we don't cure any of the animals. 15 days, we cure most of them. 19 days, which is now our standard protocol, we cure all of them. We treat it with posaconazole, really basically the same result as we saw with our compound. End of treatment, there's no parasites. As soon as we immunosuppress, all the mice rebound. All the mice but one rebound. And that's true even if we lengthen the time of treatment. It's true if we lengthen it beyond this. It's true if we increase the dose of posaconazole threefold over this, even though the PK is still increasing in a linear fashion. Whatever we do with posaconazole or with our compound, we're unable to cure mice after immunosuppression. So now it doesn't look like it's our compound. It's starting to look like it's the target. And then whilst we were in the middle of this work, data were reported from clinical trials using posaconazole and with E1224, and the data were very similar. So patients at the end of the treatment period, which I think was 60 days in both trials, were parasite-free. When they were looked at again 6 months later, without immunosuppression, obviously, but when they looked at again 6 months later, fully 60% of them now had detectable parasites. And in control arms with benzonidazole, the patients were about 90% parasite-free if they stuck to the protocol, which most of them didn't. So back to the drawing board. We're a couple of years in now and shedding people off the project, you can imagine. Right? So we thought, well, how can we find new drug targets for Chagas disease? We've had some luck in the past in both the TB program and the malaria program, taking this approach of running phenotype-based screens, raising drug-resistant parasites in the lab, sequencing the genome, and at least getting a mechanism of resistance, and in many cases, insights into the target and mechanism of action of the compounds. So we thought we would do this and apply it to Chagas disease and Leishmania and sleeping sickness. The problem was that when we ran our Chagas screen, cruzi screen, no matter how much we tried to weed out pharmacophores that looked like they were CYP51 inhibitors, we just kept finding CYP51 inhibitors over and over and over again. Eventually, we found this compound that actually came from a Leishmania screen and had quite nice activity on both T. cruzi and on the insect form of T. brucei. Now this compound, again, was pretty potent off the deck, 50 nanomolar cellular activity. We worked on all the forms of T. cruzi, both the intracellular proliferating form and the insect form and the blood form with reasonable potency. We thought, okay, well, maybe now we have a lead and we can try to figure out how this compound is working and maybe build a drug discovery program around it. So we played the same, basically the same approach that we've used in malaria and TB. It's basically the same approach, but with much more work. So we started growing T. cruzi epimastigotes in the presence of low concentrations of this compound, and we left that going for months until the parasites were growing at the same rate as the control flask. And then we upped the dose a little bit, and then we waited, and then we upped the dose a little bit more, and we waited. And after a year of patient culturing by the postdoc Shilpi Khare in the lab, we got something like a 5 to 10-fold shift in resistance of this parasite culture, which we were, not overwhelmed with. But we thought, well, it's already been a year. We better dive in. So we cloned out 3 clones from this culture. Unfortunately, they all came from the same flask. They looked like on sequencing like they're SIBs from each other. They all shared the same mutations. But one thing that was interesting that we didn't know ahead of time was whether these genetic changes that we would see would be very complex rearrangements of the genome or duplications, or if they'd be reasonably simple and easy to understand. And luckily, it was the latter. So we found 3 mutations that were homozygous, one in cytochrome B, one in a protease, one in a hypothetical protein. And then we were trying to figure out, okay, well, which of these should we go after? So we got a good clue by testing the compound on some other organisms. So here you can see the EC50s on the y-axis. So it's a bit confusing, but a low bar means potent activity. When we look at the insect form of T. brucei, we see quite good activity. When we look at the human blood form, we see quite poor activity. And this caught our attention because we know that the insect form is actually dependent on the electron transport chain for viability, whereas the human bloodstream is powered by glycolysis. So if this was working on cytochrome B, you'd expect to see this pattern. And consistent with that, although we had rather weak activity on yeast, we got much better activity when we grew the yeast in glycerol than we did in glucose. Again, when the yeast are on glucose, they can survive by glycolysis and don't need electron transport. Finally, and somewhat kind of spoiling the story, we had two strains of Plasmodium falciparum, with one of which had a substituted yeast DHODH that removes the requirement for cytochrome B, and we didn't see any shift here. And so, we thought, well, maybe it's just not active on Plasmodium, but we're going to focus on cytochrome B. So we moved on, and one of the things that we realized then that maybe we should have noticed right at the beginning of the project, but only noticed when it became very, very obvious to look at it, is that the compound that we developed as a hit molecule looks quite similar to ubiquinol, which is a substrate for, well, actually the product, I guess, of cytochrome B. Sorry, substrate of cytochrome B. And cytochrome B is shown here, the secondary structure. It has two active sites, the QP site and the QN site. It affects the reduction of ubiquinol to ubiquinone, passes those electrons down the electron transport chain. And here is our resistance mutation that falls squarely in the QN site, the leucine to phenylalanine mutation. So we had more support for this being cytochrome B in the literature before we did any real experiments of our own. And one of those interesting coincidences is shown here, which is a mutation in the yeast cytochrome B that had already been reported in the literature. That's exactly the homologous mutation to the one that we found in T. cruzi. This leucine to phenylalanine, again, falls in the same site in cytochrome B and confers resistance to yeast cytochrome B inhibitor. So even though there's very little identity between the two enzymes, this leucine is conserved and this resistance mutation was conserved. So then we thought, okay, well, how can we try to build this case? And one of the ways to do that is to say, well, what if we take independent inhibitors of the same target and ask whether we can confirm the essentiality of cytochrome B in T. cruzi by testing other compounds that are known cytochrome B inhibitors. And so we picked 4, as shown here. All 4 of them had activity. What was really gratifying is that the 2 that are known to work through the QP site, to bind at the QP site, showed no shift in the evolved resistant clones. The 2 that are known to bind at the QN site saw quite a good shift, actually, when we looked at their activity on the T. cruzi resistant clones. And sure enough, when we model the yeast cytochrome B structure and layer on top of it the T. cruzi sequence, we can see a steric clash when you swap leucine. It's the leucine here that gets mutated in resistant clones, comes in here, a steric clash that should excuse me, prevent binding of the inhibitor as it's docked in this model here, presumably still allows the natural substrate ubiquinol to bind. The kind of next experiments really kind of drive this home, I think, with some actual demonstration that the compounds inhibit respiration in T. cruzi. We set up an assay here again with T. cruzi epimasticates looking at oxygen consumption, and as you titrate in the compound, you can see quite a nice inhibition of oxygen consumption with an EC50 of about 50 nanomoles, so quite similar to our potency on inhibiting parasite growth. When we look at the resistant clones, again, we see quite a good shift in inhibiting oxygen consumption in the resistant clone. And then finally, to demonstrate that we saw inhibitory activity at complex 3, at cytochrome B, we set up an assay in permeabilized T. cruzi cells, again in epimasticates, where we poisoned complex 1 with rotenone and complex 4 with cyanide, and then fed this with electron donor, decyl ubiquinol as an electron donor, and cytochrome C as an electron acceptor. And we can measure the changes in the redox status of cytochrome C through spectrophotometry. And we see quite a nice inhibition of complex 3 activity when isolated in this way with our compound of interest. And as we'd expect in the resistant clones, we see a shift of about a log in the ability of the compound to inhibit cytochrome C reduction. So let me just summarize this part of the talk before I move on. So we have started off with a program that would say we'll take all comers, we'll figure out, let the phenotype screen, let us know where best to work. Our answer came back as CYP51. We worked on CYP51. It works beautifully in vitro. It works great in the acute model. It doesn't work. I mean, I keep saying it doesn't work at all in a chronic model. It does knock down parasite load at the end of treatment, but none of those mice are cured. They all come back after immunosuppression. We went back to the drawing board. We ended up with hits that came out of our Leishmania screen. We focused on one of them, which I've shown you here, 7686, through resistance generation, and then follow-up in functional assays. We're pretty sure that this is working through cytochrome B inhibition. I can tell you that we've developed analogs of this compound now that have good PK properties and should give us enough coverage to test whether this target is useful in vivo. And those experiments are starting this month. But we're pretty confident that this is going to work out. We're quite excited about this. And then another point which I think is worth underlining is that the target identification approach of resistance selection and genome sequencing, although it's very well worn in many other fields, hadn't actually been applied to kinetoplastid drug discovery before. We didn't know whether we'd be able to get a simple enough pattern of mutations to make sense of it. And it did work. It took a lot of culturing, but it did work. And it wasn't a one-off, and we've done this now with, depending on how you count, 3 or 4 other pairs of lead compounds and targets with biochemical activity and so on. So I think this is a generalizable approach for kinetoplastids, as it's been for other infectious diseases too. Okay, let me just check the time. Okay. So I'm going to go on to Leishmaniasis for the next 5 or 6 minutes. Leishmaniasis is a disease that actually has about a million cases per year, which is a tremendous burden. Most of those are cutaneous leishmaniasis, which is lesions on the skin, typically self-healing, but often scarring. And particularly in the New World, those lesions, those cutaneous leishmaniasis lesions can advance into mucocutaneous leishmaniasis, where the infection spreads into the mucous surfaces, which is incredibly disfiguring and can be quite dangerous. So for CL, most of the cases are in Iran, Syria, Saudi Arabia, Afghanistan, and then in Latin America, Peru, and Brazil. The focus of our program was actually on visceral leishmaniasis. It's a minority of infections, but still about 1/3 of a million per year, and causes tens of thousands of deaths each year. This disease is treated, again, with drugs that have been developed decades ago. Most of them are, well, all of them have flaws that make them inappropriate, really, for convenient use. So either they're expensive, or they're parenterally administered, and they have tox effects that require some healthcare infrastructure, or there's not good enough efficacy, or the one oral drug, miltefosine, is not particularly well tolerated. It's gastrointestinal problems, and it's teratogenic, so it can't be given to pregnant women. So there's really a hole here for us to try to contribute new drugs to the development pipeline for leishmaniasis. The life cycle of the infection is summarized here, starting with extracellular parasites, flagellated parasites in the insect gut. The insect that transmits this disease is a sandfly. Sandflies are, you guys have probably been bitten by them if you've traveled to Mexico, but they're tiny, so you don't know what's going on. You just feel these tiny little pinpricks around you. And I'm doing this because this is where they bit me when I was in Mexico. You don't really know them. The American military apparently call them no-see-ums because you can't see them. Anyway, so they regurgitate parasites into the bite wound. And then at the site of the bite, the parasites invade macrophages, start proliferating, just like the T. cruzi parasites, as these aflagellated amastogotes. Then they burst out of the macrophage and swim in the blood and then start infecting macrophages elsewhere in the body. So we thought, okay, we should try Leishmania 2. In contrast to T. cruzi, this is a kind of long and very interesting discussion. In contrast to T. cruzi, at least in Leishmaniasis, we have solid evidence from the drugs that have been used so far that a reduction in parasite burden leads to clinical benefit. Now, this is something that we take for granted for all infectious diseases. It's never actually been demonstrated for Chagas disease. There's a trial ongoing that hopefully will show that, but it's really an act of faith that going after the parasite in T. cruzi is going to help the patient. In Leishmania, we knew it with much more solid data. So we took an approach where we screened for growth inhibitors of Leishmania donovani, one of the causative agents for visceral Leishmaniasis. For technical reasons, we decided not to look at proliferation within macrophages, and instead we tricked the Leishmania parasites following a protocol that had been published in literature to de-differentiate without invading host cells, such by dropping the pH. And they form things that look like amastogotes when you look closely at them, and as people have done, there are many differences in metabolics and gene expression profiles, but they also share many similarities. For our secondary screen, though, we do look at proliferation within macrophages, primary mouse macrophages, and we optimize our SAR based on that. And then I'll show you we've set up a model of a mouse liver infection. So this is the characteristics of the axenic amastogote model. You can see that the axenic amastogote proliferation responds well to one of the frontline drugs, amphotericin B, with the same potency that you'd expect. This is what the proliferation looks like in macrophages. In this image, these are primary mouse macrophages. You can see the tiny nuclei around the main host nucleus there. And although the graph below is in THP1 cells, which is a bit cheating a little bit, but we see the same thing in mouse macrophages. Again, you see quite nice responses to frontline drugs like paromomycin. So this looked like a good in vitro model to drive the SAR. And then we had a mouse model that we set up with the help of Simon Croft at the London School, where we infect mice with Leishmania donovani. They get a very nice parasite expansion in the liver, and that responds nicely to miltefosine, which is our control compound. It's the only oral frontline drug that we could use as a control, and you see quite a nice decrease in parasites. Excuse me. Okay, so I'm not going to go through the drug discovery process and the optimization of the lead compound that we have in this program, but I'll just jump straight to its properties. Unfortunately, I can't show you the structure of it, but hopefully you'll forgive me for that. So this compound has quite good potency, 20 nanomolar or so potency on Leishmania species that cause both visceral Leishmaniasis and that cause cutaneous Leishmaniasis. We're also very pleased to see that it has similar potency not just on Leishmania species, but also on T. cruzi and on T. brucei. So it seems like a kind of triple threat compound. And I'm going to go through the efficacy data that we have for this compound before I wrap up. So this is the mouse model of visceral Leishmaniasis that I just described for you. You can see a slight increase in parasite load in the liver between when we start dosing and at the end of dosing when we treat animals with vehicle, but not a massive expansion of parasite load. When we treat with miltefosine at a dose that we pick to match the human clinical exposure, but in this case in the mouse, we can see a reduction of about 95% of the parasite load, which was about as good as had been reported in the literature at that point. Now, when we use our compound, which I guess I can call CD1, at very low doses, 0.3 up to 10 mpk, we can see this fantastic, well, maybe I shouldn't say fantastic. Hopefully you think it's fantastic. Reduction in parasite load all the way down below the limit of quantitation, which, as far as I know, has not been reported before for any compound, actually, frontline drug or otherwise. So we're really quite excited about this, in particular that it works at such low doses and can drive parasite load so low. We then went on to test its ability to work in a cutaneous leishmaniasis model. Here we're infecting excuse me, mice in the footpad with a cutaneous leishmaniasis strain, Leishmania major. In the vehicle arm, you can see that we get an increase in footpad swelling between infection, then start of treatment, and then after the treatment. When we treat with miltefosine, there's barely any reduction in our hands in this swelling. And when we treat with our compound here, you can see that at the start of treatment it looks roughly the same, but at the end of treatment, quite a good reduction from the vehicle in the footpad swelling, almost all the way back to baseline. And again, quite potent, 1 mg per kg or so effective concentration for efficacy. And finally, I mentioned to you that this compound had activity on T. cruzi, and I wanted to show you at least some success that we've had in the chronic infection model. I'll walk you through this figure. So here are the groups. We have vehicle-treated groups. Benzonidazole at 19 days, which I've shown you before, cures all the mice even after immunosuppression. A 10-day treatment in our hands here does not cure all the mice, and so we use this as a benchmark to see if we can improve on benzonidazole as a combination and potentially come up with a treatment-shortening therapy combination. And then our compound for 10 days as a single agent, and then in combination with benzonidazole as a single agent. So at the end of treatment, you can see that with benzonidazole, most of the mice are below the limit of quantitation. With our compound as a single agent, although at a reasonably high dose relative to leishmaniasis models, most of the mice are below the limit of quantitation, but some of them are not. And then the key differentiation comes after we immunosuppress. So benzonidazole for 19 days, as it always does, cures all the mice, even after immunosuppression. 10 days isn't enough. Most of them rebound for parasites. When we use our lead molecule as a single agent for 10 days, it actually looks a little bit better than benzonidazole as a single agent for 10 days, which we were pretty pleased to see. And then most excitingly, when we combine the two compounds for 10 days, we can cure all the mice even after immunosuppression. So we're very excited about the potential for this compound. It's moving into preclinical tox testing now. In fact, it's in the middle of that, and we're hoping that it'll make it through into clinical development. So to summarize, what I hope I've been able to convey to you today is that we, as well as other groups around the world, are now starting to look at drug discovery on kinetoplastids with modern drug discovery tools, with kind of purpose-built programs designed to find compounds that are optimized to treat these indications. We've had some success, at least in convincing ourselves not to work on CYP51 and in identifying cytochrome B inhibitors that we think have good potential. And then this last compound that I think has potential not just in one disease, but potentially in all 3 diseases. So with that, I'll go to the acknowledgement side. There's, in many pharma projects, there's many people involved. I'm not going to go through every name, but I do want to point out Feroz Supek, who runs the biology group for the Kinetoplastid program, and his postdoc, Shilpi Kare. Both these guys were trained at Berkeley, so you can imagine that the brains of the operation is coming from here. And as Mike told you, I was trained at Stanford, so I'm no help whatsoever. And Adi Nagel, who's running the chemistry group. We had some very good and helpful collaboration and financial support from the Wellcome Trust that I just want to mention too. Okay.
We have time for questions. So, Kerry?
It's curious. Did you uncover a biologic or chemical basis for why CYP51 was cropping up so frequently in those screens?
Well, I mean, the short answer is no, but I'm happy to speculate if you like. I mean, I think that, I mean, it's not an experiment that's unique to us. Other people have had this same problem or phenomenon. It looks like anything with a nitrogen that can chelate the metal ion in CYP51 is a good chance of working. It also looks like that groove is pretty promiscuous and will accept a lot of structural diversity into it. And it also seems like that CYP51 is a particular weak spot for T. cruzi growing in culture in the conditions that everybody is using. And also probably a weak spot for cruzi that's proliferating in an acute setting in animals. But it seems like there's this reservoir of parasites. Maybe they're proliferating at a lower rate, I don't know. But in a chronic infection under immune pressure where CYP51 is just not working. Why we keep finding so many compounds, you know, we don't really know. It's annoying though, because we have to weed through a lot of rubbish to, I shouldn't say rubbish, weed through a lot of compounds to find something new.
So I have a question. So what happens next? You mentioned that the last compound, the CD1, is going into toxicology. You're hoping it gets into clinical studies. How is that going to proceed? Is that all going to be within Novartis? Is it going to need to continue to have support, for example, from the Wellcome Trust that you mentioned?
Good question. So we're no, I mean, at least the first clinical entry, Novartis are going to take that forward. I don't know until we see the tox profile which indication will be the first clinical entry. Obviously, if you go into CL or chronic Chagas disease, those patients have a benign infection, essentially, so they will tolerate nothing in the way of adverse events. For visceral leishmaniasis or sleeping sickness, you know, there's much, I mean, you don't want to give them something poisonous, although actually frontline drugs are potentially poisonous. But, you know, that might be a greater tolerance for a compound that might have some warts on it. So at the moment, we don't know exactly where it'll go, but Novartis have committed to characterizing the compound through tox, and if it's clean enough to do at least a proof of concept trial in one or other indication.
Richard, that was really lovely. I have a couple questions, and mostly demonstrating my extreme ignorance. So to follow up just on the CYP51, what can we learn about the biology that inhibiting that doesn't matter in this chronic phase? That's question number one. Yeah. Second question is maybe I missed this, but do we know what's going on with that last compound that you talk about that works on leash and T. cruzi, and does it work on brucei?
Oh yeah, let me do the second one because it's much easier, if that's okay. So the last compound does work on brucei and leishmania and cruzi. The cytochrome B inhibitor was the one that didn't work on the human bloodstream T. brucei form, and that's because those parasites are able to survive on glycolysis alone. The insect form of T. brucei actually uses electron transport, and we see, you know, some good, not great, but some activity on that. So in terms of what we can learn about the biology of T. cruzi with the CYP51 inhibitors, you know, similar to the answer I gave before, I don't have any good answers for you, right? I mean, I can only guess. I presume it may be a little bit like TB. I presume that there's some dormant reservoir of bugs in a chronic setting, potentially under immune pressure, David, that has changed the sensitivity, and I don't know why. Maybe just because they're proliferating less, they need less ergosterol. You know, we already treat with benzonidazole, for example, which we know when we've looked at time courses of parasite reduction in an acute infection works quite quickly. But we know that in a chronic infection, we have to dose for 20 days. And if we dose for 10 days, it's not enough. And although I think the logic behind the human clinical dosing schedule for benzonidazole is somewhat lost in time, at least I'm not aware of it. Those patients are treated for 60 days. I mean, that's a really long time. So there does seem to be some sort of stubborn population of parasites. Now, this has got nothing to do with your question, but I think it's an interesting point, if you'll indulge me. So what we don't know, actually, is whether if you knock down parasite loads sufficiently, but don't cure the patients of parasites completely, is that enough to have clinical benefit? Because as I told you, 70% of the patients don't go on to develop disease. And maybe if you take that 70% to 98%, you've made a big difference and patients die of something else beforehand. We don't know the answer to that. But what I do know is that the path to registration for a Chagas disease drug, if we have to show clinical benefit, is going to be long and expensive. And the enthusiasm, at least for me to try to sell this to Novartis Development, or probably for anyone, to embark on such a trial is going to be much greater if you can say, you know what, we've nailed the parasites in a mouse model, there's none left, than if you make some argument saying, well, we've knocked them down 2 logs, probably things will be good. You know what I mean? It could be the case that if you tracked patients treated with Posaconazole for many years, or if you combine with benzonaidazole, or who knows, I mean, there may be potential there, but at least from our point of view, we're looking for a compound that will nail the infection completely.
It's about I have a general question about your preclinical models for the infectious diseases. So I'm just wondering about how much similarity do you see with the preclinical models compared to clinical efficacy? And also, like, what percentage of drugs you test successfully in preclinical trials were able to be successful in the clinical scenario?
Yeah. So for Leishmania, the model we're using has been quite well validated with all the frontline drugs, not by us, but by other groups. Simon Koff's group has done a lot of that. And pretty much things that work in the mouse work in the human. So it's a bit of a luxury, actually, for us. Well, most of our projects in the Institute are not infectious disease projects where you really worry about whether you're modeling an inflammatory disease, for example, in mouse, with any degree of relevance to humans. But for infectious disease, you're killing the same bugs with the same target, and in general, you get pretty good translation into humans. So for Leishmania, we're pretty confident. For cruzi, it's a bit harder to say because there are so few data points of compounds that have been tested in people. And in fact, as I mentioned at the beginning of the talk, for no drug, including benzonaidazole, do we yet know in people that knocking down the parasites with that drug leads to clinical benefit. We just don't know. So as we don't know, it's basically impossible to say, well, if it works in the mouse, it must work in people, because we don't actually know if anything really does work in people yet, right? We know benzonaidazole will knock down parasite load in people, and we know it'll do it in our mouse model. We know that Posaconazole behaves in the same way in our mouse model, which is a kind of unique niche, right? End of treatment, no parasites, immunosuppressed, parasites come up. Looks very much like the results in the Chagas trial in Spain. End of treatment, pretty much no parasites. Wait 6 months, parasites come up again. So, so the sort of face validity, do they look the same? They kind of look the same, but there's always a risk, right?
Great. Well, if there are no further questions, let's thank Richard one more time. So that brings us to the end of our 7th symposium, annual symposium. I want to thank our volunteers and the SEND staff, especially Zalose Alliance, as well as our sponsors, Aduro Biotech, Beyond Academia, which will be a great conference coming up in March for those interested in careers beyond academia. And to, of course, Sam Wheeler, whose donation makes SEND and this conference possible. Please join us for refreshments and a poster session right in the foyer. Thank you again.